Hello everyone, what's up guys? I am Kwizera Isaac, a student from Ines Ruhengeri in Department of Biomedical Laboratory Science, Year 3, Class A. So I'm going to share this video talking about the cooperation between T and B lymphocyte and their regulation, and also the effectiveness of vaccine. So before we dive in, let me give you a little background. This video means a lot to me because it helps to understand how your body defending against harmful agents like pathogen, toxins and cancer cell. I hope you enjoy it, and don't forget to hit that like button and subscribe for more videos, and feel free to leave a comments below with your thoughts. So let us get started. Here are some topic I'm going to discuss on it in detail in this video presentation. So my task is from question number 13 to 16. So let me start with my question number 13. It was asking about how do T helper cells support B cell activation. We have seen T helper cell divided into two main parts, or divided into two main types, TH1 and TH2. TH1, we have seen that it's a produce cytokine, support T cell and macrophage activation, and also support inflammation. Also, T helper cell 2 or TH2 cell support B cell activation, and other immune response require antibodies. So T helper cells, specifically TH2 cells, support B cell activation by providing necessary signals through the following ways. We have seen that it can recognize the MHC class 2 complex on the surface of B cell through its T cell receptor. This interaction is not sufficient for full activation. So T helper cells secrete cytokines such as interleukin 4, interleukin 5, and interleukin 21. These signals help B cell to proliferate, differentiate, and produce antibodies, leading to an effective humoral immune response. Here we have diagram which explain it very well. This diagram explain very well activation of B cell. Here we have T helper cell which recognize MHC 2 complex on the surface of B cell by using this T cell receptor. But as we have seen, this interaction is not necessary for full activation. That's why we need circulation of cytokines like interleukin 4, interleukin 5, and interleukin 21 as signals. This will help B cell to proliferate and differentiate, and also to produce antibodies. The next question was asking what is class switching and how is it regulated. Actually, class switching is a process that allows B cell to change the type of antibody they produce without altering the antigen specificity. Initially, B cell produce IgM, which change to different class of antibodies such as IgG, IgA, or IgE, depending on the signals it receives. Simply, class switching is a change of IgM to other classes of antibodies. Here there is a diagram which explains it very well how this how the different classes of antibody will be formed from IgM. I hope this diagram explains it very well. So about the regulation of class switching, we have seen that the class switching can be regulated by a combination of cytokine signals, costimulatory signals, and transcription factors. So the question here was what are T follicular helper cells and why they are important. Actually, T follicular helper cell can be defined as a specialized subset of T helper cell that reside in germinocenter of lymph node and spleen. Actually, as we have seen, T helper cell play important role in activation of B cell and antibody production. So if this T follicular helper cell, if it's a subset of helper cell, means also it can play important role in activation of B cell and antibody production within just within the germinocenters of lymphoid tissue and promote affinity methylation. So T follicular helper cell, they provide essential signals to B cell through cytokine and surface molecule. Example, there's a CD40L and CD40. When there is interaction between CD40L interacting with CD40 on B cell, there will be a activation of B cell and production of antibodies. The last question was asking how or what make vaccine to be effective. So here you have to understand what is vaccine. When they say vaccine, what do you understand? Yeah, before we're talking about the effectiveness of vaccine, firstly you have to understand what is vaccine. Vaccine is a biological preparation that stimulates the immune system to recognize and fight specific pathogens such as virus or bacteria. Then vaccine effectiveness is a measure of how well vaccine work in the real world. So vaccine become effective if it induces strong immune response without causing disease. So the effectiveness also depends on several factors that ensure that it can stimulate a strong, long-lasting immune response and protect against the targeted disease. Here are some factors that make vaccine to be effective, including how vaccine is administered, because we have seen the different route of administration, like intravenous, intramucosal, and intradermal. Yeah, there are different routes of administration. So how vaccine is administered will be a factor which makes vaccine to be effective. Also, the immune response it generates and the population that vaccine tagged will make the vaccine to be effective. Here are some key elements that make vaccine to be effective, like proper antigen choice. Here there is a targeting the right part of pathogen. Vaccine to be effective, it must target the right part of pathogen. Also, the immune system activation, the vaccine should stimulate B cell to produce antibody that can neutralize or block the pathogen, as well as humoral immunity, and promote activation of T cells, which are responsible of killing infected cells. Here are some references which helps you to understand very well the cooperation between T and B lymphocyte and their regulation, and also the effectiveness of vaccine. Yeah, this is the end of my video. Thank you so much for watching this video. Thank you so much for your constant support. Your support means the world to me. I'm so grateful for your like, comments, and share. Stay tuned for more contents coming soon. Thank you so much.